Today's lecture is meant to be an overview of the immune response to just work our way into understanding how the immune system functions. Before we actually dive in, I want to address the fact that what we focus on for this particular course is considered the vertebrate immune response. We will have a lecture later on in the semester that looks at other types of immune systems other than that of a typical vertebrate. However, for the purpose of this course, we will focus mostly on the vertebrate immune system. So to begin with, the immune system evolved to protect multicellular organisms from pathogens. Pathogens are organisms that cause disease. We know that the immune system is highly adaptable. It is extremely complex in that there are numerous cell types and different types of molecules that must all interact together in a very dynamic network to elicit a complex and full immune response. But two words that I want to start off with that you will hear again repeatedly throughout the semester is that the immune system involves two key components. There is recognition and then a response. Recognition is the ability to distinguish foreign objects or potential pathogens from self-components, and this recognition is in fact essential for eliciting an immune response. Once the recognition takes place, this then triggers a response of some sort. And again, throughout the semester, we will build up our knowledge base in understanding the multifaceted components associated with the vertebrate immune system. We know that the response, in a sense or in a nutshell, eliminates or neutralizes the invader. And we also know that responses differ depending upon the type of pathogen encountered overall. So the first definition I want to provide you with is the word immunity. Immunity has a very broad definition associated with it. It is a state of protection from an infectious disease. And immunology is the study of immunity or the state of protection from an infectious disease. The entire field of immunology developed over hundreds of years. This included observations of those who had recovered from certain infectious diseases and were protected somehow from future or subsequent infection. We know that the word immunity comes from the Latin meaning immunis, which means exempt. And so overall, again, immunity is a state of protection, basically being exempt from a repeated infection from the same pathogen. What I typically like to do in my courses before I jump into the overview of what we're going to talk about is take several steps back and look at a historical perspective. This gives us at least a little bit of a taste of some of the documented works that allow us to currently understand how the immune system functions. So we can't just jump right in. I typically, again, like to start off with a little bit of background information. So the first documented case that I want to talk about was back in 430 BC by a Greek historian known as Thucydides. Thucydides wrote about a plague known as the Athenian plague. The Athenian plague occurred because individuals were living in very close quarters and practiced very poor hygiene mechanisms. Individuals with this plague had fever, red skin, coughs, violent vomiting, and black sores all over their skin. What Thucydides noted in his writings was that individuals who recovered from this particular plague could actually nurse the sick and not get sick again. And this was a really interesting concept overall. If we jump ahead quite a bit to the 15th century, we have documented cases in the Chinese literature in attempts to induce immunity in individuals. Smallpox has been a problem disease for centuries. It is a viral infection or due to a viral infection. It causes diarrhea, fatigue, fever, rash, vomiting. In addition, it does create these pox or pustule sores on the surface of the skin. Now, what was noted in the Chinese literature at the time was a technique that is referred to as variolation. With variolation, dried crust from the pox of a pox pustule would either be inhaled by a healthy individual or inserted into a cut in a healthy individual. And somehow this was noted to prevent disease in the future. And again, this was very interesting in that this was an attempt to induce immunity to this disease known as smallpox. If we jump ahead a bit, we have the work or the example from Mary Wortley Montagu. She was the wife of the British ambassador to Turkey, and what she witnessed was the same variolation technique we just discussed in the Chinese literature was very successful in preventing disease in the Turkish people. She was so taken aback by this that she decided to have her own children treated in this particular way and found that her children then again were immune to smallpox. Why this is so noteworthy is this is the first Western documentation of utilizing this variolation technique to prevent disease overall. If we jump ahead to the work of Edward Jenner. Now, Edward Jenner was a physician, and he made several observations. One of the observations he made was with milkmaids. These are girls or ladies that would milk cows that had a disease known as cowpox, which happens to be a more mild disease related to smallpox. Those individuals seemed to be immune to smallpox, and this was very interesting and perplexing to him. So what he decided to do was to inoculate a boy with the variolation technique with cowpox. He then later went to infect the same child with smallpox and found that the boy was in fact immune to smallpox. Now, in today's standards, something like this might be a little bit unethical. However, at the time, Edward Jenner noted something and wanted to test his hypothesis to see if in fact individuals could be immune to smallpox if they had been exposed to cowpox previously. Word spread about this variolation technique, but it took about 100 years before the technique was applied to other types of diseases. Then we jump ahead just a little bit to the work of Louis Pasteur. Now, Louis Pasteur was in fact a physician and also a scientist who was really interested in microbiology. We're going to talk about two specific sets of experiments that he conducted. The first one dealt with a disease known as cholera. Cholera is an infection of the small intestine and causes extreme diarrhea and vomiting. It is in fact due to a bacterial pathogen. He was interested in studying cholera. In particular, he used chickens as his study organism. What he found with the particular strain of cholera, once he grew the bacteria up and injected it into chickens, the chickens would actually die from the infection with this particular bacterium. It turns out that he let the culture of bacteria get a little bit old, and we'll talk about what that concept is here in just a second. But he took what was referred to as an old culture and injected it into the chicken or chickens and found that the chickens would get sick but stay alive. So this was really interesting to him in that a newer, fresher, more virulent culture of bacteria would cause death in the chickens. However, if he let the culture sort of sit out and become weakened over a period of time, the chickens, when infected with the bacterium, would get sick but stay alive. So something about this culture, in that it had been weakened in some way, made it less deadly overall to the chickens. What he decided to do then was take these chickens that survived and treat them with a fresh, completely virulent culture of bacteria that caused cholera, and found that the chickens would survive. And so here too, there was an induction of some type of immunity that occurred when the chickens were exposed to a weaker strain of the pathogen. He formulated a hypothesis, which is known as the attenuation hypothesis. And this basically says that you have an attenuated form of the pathogen, and attenuate just means to weaken. And in this particular case, what he proposed that the weakened strain could be injected to protect from the full-blown virulent strain of the bacterium. He decided to test this hypothesis with a totally different type of bacterium. In this case, he tested it with anthrax utilizing sheep. In 1881, he tested the hypothesis by vaccinating sheep with an attenuated strain of anthrax. And again, an attenuated strain is one that has been weakened over time. He then challenged vaccinated versus unvaccinated sheep with virulent anthrax bacteria and found that, in fact, those that were vaccinated with the attenuated strain of anthrax bacterium were able to survive a full-blown infection with completely virulent anthrax. He also later on went to try this in the first human example with a rabies vaccine. However, he still didn't completely understand how it worked, but attempted to try this with a boy who had been bitten by a rabid dog with the rabies vaccine. And this is again the first human example of inducing immunity. So the previous examples that we just discussed included. This table comes from your book and just gives you some idea of individuals, scientists that have won Nobel Prizes for their immunologic research. And by all means, this is not a complete list, but it does include several key scientists and their contribution overall to the study of the immune response. So when we talk about immunology, we have to kind of go hand in hand with a field known as medical microbiology. Now, in order to continue discussing the immune system, we have to add some additional definitions. And that is, a pathogen is an organism that causes disease. I mentioned this earlier in the lecture. Pathogenesis is the means by which the disease-causing organism causes illness in the host. When we talk about pathogens, there are in fact 4 broad categories of pathogens. There are viruses, and viruses are non-living entities that require a host cell type in order to reproduce and produce more viral particles. We know that there are a whole host of diseases associated with viral pathogens, including our novel coronavirus, which is causing the global pandemic that we are living through currently. Throughout the semester, we will reference some examples of the coronavirus and the immune response specifically to the coronavirus. However, keep in mind that this course, again, is an overview course looking at the entire immune response and not just one specific pathogen in question. Okay, so there are again 4 categories of pathogens: viruses, like I just mentioned, bacteria, which are also a broad category of pathogens that are unicellular organisms that are prokaryotic. There are a number of diseases listed here that are associated with bacterial pathogens. And then the last 2 categories, fungal and parasitic pathogens, present typically a larger problem to the immune response, and that is both fungi and parasites are eukaryotic organisms. The fact that they are eukaryotic organisms make it harder for that recognition that I mentioned at the very beginning of the lecture to occur, meaning that the cell types associated with these pathogens are fundamentally the same cell types in the host system. And so this then causes a problem with recognition. Now, it doesn't mean that the host system cannot interact and fight off fungal and parasitic infections. In fact, our immune system can, in fact, elicit immune responses against these pathogens, but these two categories of pathogens present an additional type of problem to the immune system. In addition, both fungi and parasites can come in single-celled and multicellular forms, which also presents an additional problem for the immune system, but we will talk about this a little bit later in the semester. The important thing to note here, again, is the 4 broad categories of pathogens that we will be dealing with overall throughout the semester. As I mentioned at the very beginning of the lecture, the immune response of a typical vertebrate system is an extremely dynamic mechanism. There are multifacets that are involved in a typical immune response. The 2 broad categories, though, that we will discuss are what are known as the innate immune response and the adaptive immune response. There's one third facet that we will get into and we will discuss later on in the semester, known as the complement system, and the complement system is a third component not listed on this particular slide. But let's discuss both innate immunity and adaptive immunity in general terms. The innate immune response is also referred to as the inborn or natural mechanism. It is, in fact, the first line of defense. It is always in place, does not need any specific trigger to elicit an immune response other than the recognition that we're going to talk about in just a little bit. But again, this mechanism already exists, is already ready to go to elicit an immune response. That is clearly different from the adaptive immune response, which is considered the acquired mechanism. It is a second line of defense, meaning that there has to be an initial recognition. There's a little bit of a timeline associated with it, but then a response is elicited after that recognition phase takes place. Now, the difference between the innate immune response and the adaptive immune response also comes down to non-specific mechanisms in the innate immune response. There are molecular and cellular components, again, that are present before an infection takes place, that are involved in the recognition of broad categories of pathogens. So non-specific mechanisms involved, broad categories of pathogens are identified in the innate immune response. This is very different from the adaptive immune response in that it is highly specific to the pathogen in question. The adaptive immune response develops in response to an infection and adapts to eliminate that particular pathogen. In our next lecture, we will discuss cell types that are involved in the immune response, keeping in mind that any one cell type is not necessarily just found playing a role in innate responses or adaptive responses, but there are cells that dominate in either one of these categories. In particular, for the innate immune response, we will learn quite a bit about phagocytes, which is a cell type that is involved in destruction of pathogenic components. In the adaptive immune response, the cells that play a large role are known as lymphocytes. These are things known as B cells and T cells, which again we will discuss in more detail in the next lecture, but these are the cell types that dominate in the adaptive immune response. For the innate immune response, the response can occur within hours of an infection. This is again because this mechanism is the first line of defense. It's already in place, ready to go. So if you are infected with a particular pathogen, your innate immune response kicks in within hours of an infection. However, the adaptive immune response does in fact require a few days after infection because of the highly developed mechanism of recognition that needs to take place with the specific pathogen and then elicit the immune response. So there's definitely a time lag associated with the adaptive immune response. And then the last thing that I want to point out that is not on this list, but the adaptive immune response does have what's referred to as a memory component, which is not found in the innate immune response. The adaptive immune response, again, kicks in because of the recognition of a specific pathogen. Once a specific pathogen has been encountered, there are cells that are referred to as memory cells that can recognize that pathogen upon subsequent exposure and allow for an almost immediate immune response due to the memory that has been elicited. This slide is meant to be an overview of what happens in both the innate and adaptive immune response. The very first step that needs to occur is that an infection does in fact need to take place. What typically happens with an infection is that a pathogen enters through the breach of some physical barrier, and again, we will discuss this in more detail a few lectures down the road. Once the infection has in fact occurred, the innate immune response kicks in immediately, like we just discussed, because it is in fact the first line of defense. There are several cell types that are involved in the recognition that then trigger an immune response to destroy those pathogens, but also to then trigger a connection to the adaptive immune response. The adaptive immune response is then kicked into high gear, and a complete destruction of that pathogen occurs overall. This graph is meant to drive home the point of how important the adaptive immune response is for repeat exposure to a given pathogen. What you're looking at along the x-axis is the time in days of exposure to a particular pathogen, as well as the response that occurs. So in the first half of the graph, you're looking at both the innate immune response and the adaptive immune response, with the magnitude of immune response listed in either the light blue for the innate immune response and then the darker blue for the adaptive immune response. What you can clearly see here is, again, that shift or the time lag that occurs before the adaptive immune response kicks in, but the magnitude of that response is much greater than the innate immune response. The important thing here, though, to note is upon repeat exposure to a given pathogen, the innate immune response kicks in just like it did previously, so there's really no difference upon repeat exposure to that same pathogen. However, what you can see is the magnitude of the response with the adaptive immune system is much greater with repeated exposure to this pathogen, and that again goes back to those memory cells that I was just discussing in the previous slide. And we will discuss, again, all of these concepts in more detail in lectures to come. So throughout the semester, what we are going to do is build up our understanding and our knowledge base of how the vertebrate immune system functions. I like to take the approach of sort of pulling it apart piece by piece so that we can understand it in tangible packets of information rather than just being thrown in all at once. So we will do that throughout the semester, is build up our knowledge base. Then at the very end of the semester, we will then discuss what's referred to as immune dysfunction. What happens when the immune system doesn't work the way that it's supposed to? There are broad categories of immune dysfunction. There are what are known as allergies and asthma, specifically referred to as hypersensitivity. Hypersensitivity occurs when there's a problem with recognition, in that something is being recognized as a potential threat or pathogen that is not truly a potential threat or a pathogen, and an immune response is elicited. So anyone who suffers from allergies understands what happens to them physiologically when their allergies kick in, but this again is when the immune system is not functioning the way that it's supposed to. And again, this is referred to as hypersensitivity. We will also discuss autoimmune disorders, and autoimmune disorders also are a problem with recognition. Self components are recognized as a potential threat, and immune responses are elicited against self components in the host. You can see why this would be a potential problem if self components are targeted for destruction by the immune system. And then the last broad category is known as immunodeficiency, and this is just where some or multiple aspects of the immune system do not function at all, and it is distinct from both hypersensitivity as well as autoimmune disorders. So again, hopefully you've enjoyed this sort of snippet of information to introduce the concepts, give us a historical perspective, and in our next lecture we will dive right in to discuss cell types involved in the immune response.